our introduction? Oh.
Absolutely. Well, Tarek, can you hear me?
Yes.
Great. Well, I can see Jonathan. Good. So welcome everybody to the CVRTI Distinguished Speaker Seminar Series. Unfortunately, I cannot be with you because I have to quarantine due to COVID, but the good news is that I feel fine. So it gives me great pleasure to introduce today Jonathan Kirk from Loyola. So Jonathan is an associate professor and vice chair of the Department of Cell and Molecular Physiology at Loyola, and also the director of the Loyola Proteomics Core. Jonathan completed his undergrad in bioengineering at the Washington State University, and then he performed his PhD studies at the Department of Bioengineering at the University of Pittsburgh. He then moved on to perform a postdoc at David CASA's lab in Johns Hopkins. And during his time with David was probably the first time that he had some interactions with investigators here at CVRTI through some collaborative projects that we discussed a little bit earlier today in our visit with him. So his lab uses sophisticated biophysical assays to study human and animal models of disease to understand the molecular mechanisms that cause the sarcomere and the myofilament to malfunction. So he's interested in heart failure and cardiomyopathy, and using these proteomic techniques such as mass spectrometry to identify the proteins that they are involved, and some of these the abnormalities that can be reversed to attempt to treat the disease. We are very excited that he's with us, and he's going to share with us some of his research through this seminar. Thank you, Jonathan, for visiting us in person. And again, sorry, I cannot be there in person.
Really understandable. Thank you very much for the invitation. I'm really excited to get back to in-person visits and in-person interaction. I personally really enjoy giving a seminar to actual people, and you can't turn your camera off and I can actually see your faces. So thank you for that kind introduction. And I'm going to talk to you today about a recent project of ours looking at a protein called BAG3. So we're going to talk a lot today about BAG3, but from a larger sense, what we're interested in here is a question that's kind of been in the back of my head for many, many years throughout my training, which is the sarcomere. Let's see if this works. Oh no, it stopped working again. All right. That's not working at all. Cool.
Maybe the car stopped.
Yeah. Okay, sorry. So here are my disclosures. Yeah, there we go. Just need to click on it again. All right. So the concept here, right, is that the sarcomere, so the actin and myosin mostly, but all these other accessory proteins, are the engine essentially for the heartbeat, right? So the actin and myosin interaction, forming of cross bridges, that's what's actually causing the heart to beat once a second, every second, more or less. And so these proteins are under constant mechanical stress. They're constantly being pulled and pushed together as they're generating force. And yet the cardiomyocyte, terminally differentiated, has to be able to remain operative the entire cycle, the entire life of the organism. And so how do you keep this sarcomere structure operating in peak physical condition the entire life of the individual? And it sort of raises this question of how are these sarcomeric proteins turned over, right? How do you replace pieces of the sarcomere with new pieces when you can't stop contracting ever? Right, like, and so the title of the talk was repairing the car engine when you can't stop. The idea of the sarcomere as the engine, and how does this happen? And so when I was thinking about this conceptually of sort of how repairing the thing without stopping, this made me think of the 1994 action movie masterpiece, Speed. So starring Keanu Reeves and Sandra Bullock. If you haven't seen it, it's okay, I'll explain it to you because it's a really simple 90s action movie. The idea is that a bad guy, Dennis Hopper, who's great, puts a bomb on a bus, and if that bus slows down below 50 miles per hour, it blows up. And so this is kind of conceptually similar, right? You can't slow the sarcomere down. It's got to keep going at whatever pace the organism needs to survive. But you've got to be able to get all those people off the bus to save them. And well, of course, in our analogy here, replace them with new proteins, which doesn't happen in the movie. But regardless, it sort of sets up an analogy here for how you do this. And so I personally really like movies. And so I'm going to tell you the story today about BAG3 and sarcomere protein quality control, like we're going through the 1994 movie Speed. And so any good action movie starts off with setting up the stakes, right? So what's at stake here if this doesn't work out? And the question here is, what happens then when you don't perform adequate sarcomere protein quality control, or if you don't turn over these elements of the protein structure that generates force? So our story here goes back 50 years. It's going to start with the venerable Eugene Braunwald, who published this study in Circ-Res in 1967, back when a Circ-Res paper could be like 3 figures. And he used a cat model of congestive heart failure and essentially showed here in this figure from the paper that the muscle, the myocardium, was significantly weaker in the animals that had heart failure. Now, today, this probably isn't that startling, but this is an important finding back 50 years ago. And here it says it, that there was an extreme depression of the intrinsic contractile state of the muscle. So heart failure is fundamentally a weakening of the heart muscle itself, a systolic reduced ejection fraction heart failure. And the sarcomere is at the center of this, right? If there's a contractile deficit in the sarcomere, in the muscle, the motors that drive that contraction are going to be playing an important role at the very center of that, of course. Now, for anybody who studies anything else in a sarcomere, in the cardiomyocyte, I'm not saying, of course, that the sarcomere is the only thing contributing to the contractile state. Of course, it requires calcium activation and handling in order to generate force. It requires ATP and sufficient mitochondrial function in order to power that. And other things in the myocyte, like microtubules and protein aggregates, can interfere with the sarcomere's ability to generate force. However, if you see all of these elements, they're converging in on the sarcomere here as the primary contractile apparatus inside the cardiomyocyte, and this must be playing a fundamental role in why the myocardium itself is weak in heart failure. Before we go on, I want to talk about how do we measure sarcomere function. I'm about to show you some past data and then our own data, but I want to talk about how this is done so you understand and can interpret the data I'm going to be showing you. What we do is we take individual cardiomyocytes and we skin them, which sounds like a very violent process, but it's really just bathing them in Triton for 20 minutes. And that permeabilizes the membrane. And the reason we do that is because now we can activate that sarcomere directly by controlling the amount of calcium that is binding to troponin, moving tropomyosin out of the way, and allowing force production. We then take those individual cardiomyocytes and we pin them to a force transducer on one side here, and a high-speed length controller on the other side, length being a very important aspect of sarcomere force production, although I'm not going to talk about it very much here. We just set it to a certain sarcomere length. And then what we do is in the bathing solution around the sarcomere, sarcomeres, we start to introduce increasing levels of calcium, which activate the myofilament. And as you can see down here, as we get more and more calcium in the activated in the sarcomere, we get more and more force production, which the force transducer registers. And at some point, no more calcium doesn't produce any more force, and we get this saturating curve like this, which we can then fit to a hill curve like that. And that hill curve gives you relatively important information about the functional status of that sarcomere. One of those is Fmax, which is essentially just the maximum force that the sarcomere can generate. You can also think of it as the force generating capacity of a cardiomyocyte. The other one here is the calcium sensitivity, or the leftward-rightward shift in it. Today, we're going to talk primarily about Fmax, or the maximum capacity for the sarcomere or the cardiomyocyte to generate force. And it was first found here in this paper about a quarter century ago in Circulation from Peter Tatum's lab in a rat model of heart failure that Fmax was depressed with heart failure. And so you can see here in this top curve and in this bottom curve, which are just different models, that the black circles in both of them have a significant depression in Fmax with heart failure. Okay, just like Eugene Braunwald had shown in a twitching myocardium, the muscle and the myofilament is fundamentally weaker in heart failure. And since this original finding, like I said, in 1997, this has been replicated over and over again. So here's a study from Ken Campbell's lab at Kentucky showing in human cardiomyocytes depression in Fmax by about 40%. Here's a study in a pig model from Jolanda van der Velden's lab showing a decrease in an Fmax in this pig model of MI-induced heart failure. Here's two mouse models of heart failure, both TAC and MI-induced, showing those bottom two lines are both about a 40% reduction in Fmax. And then my own work previously in a dog model of tachypacing-induced heart failure, you can see this 40% reduction, black line compared to the red line. Ignore the blue one for now, because that's a different study. So you see over and over again, this roughly 40% reduction in Fmax for the past quarter century, leading into the findings from Dr. Braunwald. Now, despite the fact that this has been shown over and over again, the mechanisms about why Fmax are changed still aren't understood. We wanted to sort of address this. So we used our own cohort of humans, human hearts. And so I run the cardiovascular biorepository at Loyola. And therefore, we have explanted hearts from patients getting transplants at Loyola Medical Center, as well as rejected donor hearts from the Chicago area procurement network. And this is the basic demographic information from the human hearts that we used in this study. So we have nine non-failing and 21 dilated cardiomyopathy, or essentially non-ischemic heart failure samples. And you can see basically they're identical, they're the same overall demographics, except for, of course, the ejection fraction being pretty low in the end-stage heart failure group. Just so you know, whenever I show you any of data that we collected from my lab, it's going to be in all sorts of different species as we move throughout this. If you're ever confused, or if I forgot to say what species it's in, if you look up here in the upper right-hand corner, that'll indicate what species this particular study was done in. So this is in humans. If you're watching this from Zoom at home and my picture is up there, that doesn't mean the studies were conducted in me. Okay, so here's our study: 9 non-failing, 21 non-ischemic heart failure. And you can see here when we did these force calcium relationships, we see this 40% decrease in Fmax. And here, when it's quantified, this is the actual Fmax value, we get this 40% reduction in these human heart failure samples with a pretty nice p-value. Of course, I just told you that's been known for a quarter century. So this isn't a particularly novel finding, but it was important for us to set up that we could see this phenomena and now study it. Like I said, this question is why. It's not clear why Fmax decreases, the force generating capacity. One hypothesis that has existed for a while is that oftentimes in end-stage heart failure, you also see significant myofibrillar disarray. And so here's electron micrograph from a control dog, healthy dog, and then this is from one of my previous studies. And here is at another dog that had a tachypacing-induced heart failure. And so in this heart failure group, when you look at the myofibrillar structure, you see loss of Z-disc structure, you see a loss of nice digitation between the thick and thin filaments, all of this making it pretty easy to believe this would not be able to function as well as a control healthy myofilament would. And one hypothesis for why we have that structural disarray is that there is a lack or loss of normal protein quality control mechanisms in the sarcomere. The problem is the normal protein quality control mechanisms in the sarcomere aren't known. There are hypotheses of some of the involved pathways, but for the most part, how protein quality control is performed in the sarcomere is a black box. But if we consider some of the known pathways here, we can see that there are calpains that break down individual protein monomers, chaperones that bring out ubiquitinated misfolded proteins. They get either taken down by the UPS or the autophagy system, normally speaking. And the hypothesis would then be that in heart failure, you have increased stress on the sarcomeric proteins and a decrease in the pathways that clear those out. And therefore, these proteins begin to form misfolded protein aggregates or stay embedded in the sarcomere lattice themselves, and thus cause myofibrillar disarray. The structure doesn't look as clean and as nice as it used to, and that could possibly lead to disrupted function. But like I said, that's not really known. So what we wanted to do is we looked in our non-failing and heart failure samples and said, first of all, is there any indication in these that there's a decreased protein quality control of these sarcomeric proteins? When you're studying the sarcomere, it's a pretty nice organelle, I guess, or the cytoskeleton, because it's almost crystalline in structure. I'm a bioengineer by training, and so that very clean, crystalline structure and motors appealed to me as an engineer more than all those squishy, weird shaped cells. Okay, so because it's crystalline in structure, it's very easy to enrich your sample for the sarcomere because all those protein structures bind very strongly to each other. It's easy to spin everything else out and wash it away, and you're just left with the sarcomere fraction itself. So here, when I do these type of experiments, we call them myofilament enriched. Of course, it's not a perfect enrichment, but because that sarcomere structure is so huge and so high affinity for each other, it's pretty easy to remove most everything else from your sample. The hypothesis here that we wanted to first test was that if you had a decrease in the protein quality control mechanisms of the sarcomere, these proteins would be marked for ubiquitination, marked for degradation by ubiquitination. And because they weren't being cleared out, they would accumulate increased protein ubiquitination in heart failure. And you can see here in these figures that in fact that's what we saw. So about a 40% increase in sarcomeric protein ubiquitination in our heart failure samples compared to healthy non-failing samples. But that doesn't tell us that much, right? We wanted to know, where are these ubiquitinated proteins? Are they forming protein aggregates, which is also known to occur in heart failure, or are they staying embedded in the sarcomere lattice itself? So we did super resolution microscopy on these human myofilaments. And what we did was we're just looking at myofibrils here. So we're washing away the cytosol, the other possible protein aggregates. We just want to know, are these ubiquitinated proteins staying embedded in the lattice itself? And what we found here was that looking at myofibrils like this, there's a strong striated pattern of the ubiquitin antibody and staying sort of embedded in a sarcomere lattice framework. So this suggested to us that these ubiquitinated proteins that aren't being cleared out are staying embedded in the sarcomere lattice itself. But what proteins were these? We wanted to know that is, well, okay, are these actually sarcomere proteins? Are they important for contraction? What are they doing? So what we did was we took non-failing and heart failure samples. So again, these same hearts. Over and over again, we're using these same 9 and 21 hearts so that we can match up all this data and say this is happening in the same person. We did mass spec on this. We do a myofilament enrichment to start off with to say we're just going to look at myofilament proteins. We digest them with trypsin, so we end up with peptides. When you take and digest a protein up with trypsin, it actually cleaves the ubiquitin moiety off and leaves a diglycine-lysine residue behind. So you can actually use a diglycine-lysine antibody to enrich for ubiquitinated proteins. Actually, ubiquitinated peptides specifically, you can actually get the individual residue. And what we found was that there was a lot of protein ubiquitination increased in our human heart failure samples. And these were primarily on sarcomeric proteins like myosin light chain, filament C, tropomyosin, actinin, even down here, myosin, the other light chains, actin, sorry. So overall, these are a lot of the core components of the sarcomere that make up the actual contractile apparatus of the sarcomere, building up ubiquitination specifically in heart failure. And again, as you would expect in normal hearts, it's quite low, that these are being turned over more aggressively. Okay. So we come to the stakes here of this is what happens when you don't turn over the protein, the sarcomere proteins adequately. They build up, they cause dysfunction. You have a 40% decrease in Fmax, an intrinsic depression in myocardial function, and heart failure. Okay, so again, we've associated these things. Or the bus we were talking about, of course, they drive to an airport and crash into an airplane because it's a 90s action movie. Okay. So now let's talk about the mechanism here and get a little bit deeper into it. And so in any good action movie, after you establish the stakes, you've got to introduce your hero. And in Speed, it's Keanu Reeves, pre-John Wick. And in case kids today, that's all they know him from. And in our scientific story, it's going to be Beclin-2 associated anthanogen 3, or BAG3. What is BAG3? So BAG3 is a co-chaperone that has been identified in many autophagy pathways previously. And it is a co-chaperone because it binds to lots of different things, and it has domains that bind to lots of different proteins that involve lots of different signaling pathways. So we're going to go through all those in a second. It is highly expressed in striated muscle, cardiac and skeletal, and the central nervous system, and also interestingly in tumors. So we're going to come back to that a little bit later on, but it is very high in especially very metastatic cancers. So going through the BAG3 structure here at the N-terminus, we've got domains that bind to TSC and are involved in cell adhesion. We've got these IPV domains that bind to the small heat shock proteins, HSP8, 6, and 5. We have a PXXP domain that binds to dynein. It walks along dynein and microtubules to transport things around the cell. What it gets its name from is the BAG domain here, BAG3, which binds to Beclin-2, as well as HSP70 to be involved in a lot of autophagy pathways. So you can see it does a lot of things in the cell. And one of the things that we're going to be interested in here is sort of teasing apart one of those roles. The function of BAG3 in striated muscle was sort of first introduced or conceptualized here in this 2006 paper, where they did a heterozygous knockout of BAG3 and showed that in the diaphragm, the sarcomeres formed perfectly fine. So here on E18.5, that the mice had perfectly formed sarcomeres. But as soon as those sarcomeres were put under stress, as soon as the animal was born and actually had to breathe on its own and do something, when you didn't have BAG3, they fall apart. So you lose structural integrity very, very rapidly. And by day 14, you can see here, it's impressive that that muscle does much of anything at all. So this is the original suggestion that BAG3 might be important in a striated muscle, somehow controlling or contributing to maintaining a structure. And then Jörg Hofeld's group in 2010 showed that in skeletal muscle, it localizes to the Z disc with alpha-actinin, and that it could chaperone proteins out of the sarcomeric or cytoskeletal matrix into the cytosol. And this was an assay he called a release assay, basically taking enriched cytoskeleton samples here on the left, and then exposing them to high levels of exogenous BAG3. And you can see here, he just looked for the protein filament C. And thus in the treated group here in these two lanes here, it was releasing filament C from the lattice structure. And so from this, they interpreted that BAG3 could act as a chaperone for certain proteins in the cytosol, in the cytoskeleton, and BAG3, filament C was the only one they identified. And then sort of to take, finish up the history lesson here, Hashia, Circres 2010 showed in neonatal myocytes sort of a similar thing of what has been seen in the diaphragm, that you get rid of BAG3, here shown on the right, and the sarcomere structure starts to fall apart. You lose the ability to maintain normal sarcomere structure with stretch here. So again, it has to be under some sort of stress in order to uncover this. Now, since those original mechanistic studies trying to understand what BAG3 was doing, it's turned out we've discovered that BAG3 is really important clinically as well. This was a study from a large European consortium published in 2018, where they were looking at BAG3 mutations, and it's associated with dilated cardiomyopathy. And they essentially found here, they identified, I can't remember how many it was, like 138 patients or something like that. And this is the survival curve showing that they end up with only about 40% survival by about 15 years, mean follow-up after this, after the BAG3 mutations were identified. And you can see here on the right here that by the time these patients hit 50 years of age, almost 90% of them have a dilated cardiomyopathy phenotype if they have a mutation in BAG3. And this has been borne out by animal models. So Ju Chen has done some work in recreating some, very few, only one or two, of these mutations in animal models. And this one is the E455K, which is in that BAG domain. And when this mutation happens, you can see here in the red line, the homozygous mutations die quite rapidly. So all this is to indicate, yes, BAG3 appears to be super critical clinically in these mutations, but also in non-genetic heart failure. So this paper from 2014 from Art Feldman's group showed that in non-genetic heart failure, there was a significant depression in the levels of BAG3 in the cell. Okay, so all these cases, we're losing BAG3 in heart failure. All right, let's come back to our study in our work here. So in those same human samples that we used functionally, we looked at their level of BAG3 overall, and we found about a 50% reduction in whole tissue BAG3. When we just take the myocardium and grind it up and look for BAG3, we see about a 50% reduction, which isn't quite as significant as what Art Feldman had shown previously, but still pretty significant. But again, we're not interested in what BAG3 is doing in the entire cell. I mean, us personally aren't interested. What we're interested in is what it's doing at the sarcomere. We wanted to know whether or not BAG3 was actually at the Z disc binding to the sarcomere, as it had been shown to do in skeletal muscle. So here is immunofluorescence of alpha-actinin at the Z disc of the sarcomere, and BAG3 co-localizing with it, suggesting BAG3 is in fact at the sarcomere. So we did a myofilament enrichment of our human samples. This is again human, and we looked at BAG3 levels at the sarcomere specifically in those same samples. And you can see here, we see about a 25% reduction in sarcomere-bound BAG3 in these same patients where we found a 50% reduction overall. Our interpretation of this is that the BAG3 portion or localization of BAG3 to the sarcomere is so critical, the cell is trying as much as possible to maintain that level, despite the fact that overall protein levels have decreased by a much larger amount. Of course, that's just our interpretation because I think the sarcomere is so important, so I figure BAG3 must think the same thing. But let's go back to that functional data for a second. So I showed you this data, and if you remember when I actually showed you all of the actual Fmax data, there is a large spread in that. Some of those heart failure patients had what looked like pretty normal Fmax values, and other patients had really, really low levels of Fmax. And that's what we did on a patient-by-patient context. We took a look to see whether or not there was a correlation between the force-generating capacity myocytes had from those patients and the level of sarcomeric BAG3 that they had. And we found that in those patients that had preserved levels of BAG3 at the sarcomere, they had almost normal function. And as those patients that had worse and worse depression in BAG3 at the sarcomere, they had worse and worse function of their sarcomeres, of the myocytes from them. Now, I told you the other index of sarcomere function is calcium sensitivity, that leftward, rightward shift. Whereas the role that Fmax plays, or how Fmax decreases in heart failure, isn't well known, calcium sensitivity is. It's pretty well described that troponin I, myosin-binding protein C phosphorylation is altered by pKa phosphorylation and changes in beta-adrenergic signaling. That's all pretty well known. And what's been shown is that due to a decrease in pKa phosphorylation of these sites, you get an oversensitization of the sarcomere in heart failure. And that's what we saw in our patient cohort. But because those mechanisms are known, we would hypothesize they would be unrelated to the level of BAG3, right? And indeed, that's what we found. So Fmax is predicted by BAG3 levels, but calcium sensitivity, which has a known different mechanism, is not. This also suggests that this association between BAG3 and Fmax is not merely a they were sicker sort of thing, because other elements describing how sick they are not correlated with BAG3. I'm gonna take a little bit of a side here, and that data that I showed from the European consortium looking at BAG3 mutations, they actually found that there was a really interesting sex difference in patients that had BAG3 mutations. And as you can see here, in blue are male patients, and in red are female patients. And compared to the females, the males had a much worse prognosis if they had BAG3 mutations than the females. Females were protected somehow from as worse a phenotype with these BAG3 mutations. We wanted to see whether or not our own data bore that out in non-genetic heart failure. And what we found was that in our, this is actually from a different study, so this is an entirely different cohort of patients. But what we found was that male heart failure patients had very depressed BAG3 levels of the monofilament, whereas females did not. And if we took the overall levels across all of the patients, heart failure, not heart failure, or whatever, we found that females generally had a higher level of BAG3 at the monofilament than males, thus suggesting that if these mutations were affecting males more significantly, it might be because they had a lower amount to begin with and thus had less ability to deal with a genetic mutation, especially a heterozygous mutation. We wanted to know whether or not this change in monofilament BAG3 expression was estrogen dependent. What we did here, we used a rat model where we let them age out to 18 months, did OVX, and then either introduced estrogen again or not to see whether estrogen had the effect on BAG3 levels. And we found that there was not a significant change in monofilament BAG3 with estrogen. Therefore, likely it's something else, which is fine. I feel like a lot of scientific studies say females are just males with estrogen, and that's not true. There's a lot more differences than just the presence or absence of estrogen. So in this case, it's suggesting something else more complicated is happening, regulating BAG. However, even though the level of expression or level of localization of BAG3 is sex dependent, what BAG3 is doing at the sarcomere isn't. So if we pull out this association between BAG3 and Fmax in our male and female patients, we find that the functional consequence of having low BAG3 is the same in males and females. So functionally what BAG3 is doing is sex independent, even if its level of expression and localization is sex dependent. So just sort of an aside, sort of side, I don't have that all answered yet, but I think it's very interesting, and it correlates and agrees with the larger clinical data as well. Okay. Now, usually once you introduce a hero, they get knocked down, they get into trouble. And so here in Act 3 of our action movie, we're gonna knock down our hero and we're gonna do a genetic knockdown of BAG3. Obviously, all these human studies are great for showing translational relevance and clinical relevance, but it's all correlative, right? There's lots of things happening in heart failure that we're not taking into account. And so let's be more specific here and look just at BAG3. All right, this is a constitutive cardiomyocyte specific knockdown of BAG3. We do have an inducible model now, but we're just starting experiments. So this shows that you get about a 50% knockdown in the heterozygous knockdowns of BAG3. You can do this as a homozygous knockdown, but they die pretty rapidly. And so you can't really, they're not great for long-term experiments. I am going to show you one piece of data in the homozygous later on, but generally speaking, it's not a great model to use. And you can see here that the heterozygous ones over time will eventually develop sort of the dilated cardiomyopathy-like phenotype. But of course, we're interested in sarcomere. Let's bring it back. So we see in these heterozygous knockdown mice of BAG3 at the monofilament, we have about a 25% knockdown of reduction in BAG3. This is really interesting to us because remember, in our heart failure or human heart failure samples, we saw 50% reduction in overall BAG3 and only 25% reduction in sarcomere BAG3. And here in the heterozygous knockdown mice, a 50% overall and a preservation of that at the sarcomere, so only a 25% reduction. And so quantitatively, the human and the animal model here agree in the amount of BAG3 loss that we have, both overall and at the sarcomere itself. In these knockdown mice of BAG3, we see about a 40% increase in myofilament ubiquitination, similar in magnitude as what we saw in the human heart failure, suggesting that with a loss of BAG3, the sarcomeric proteins aren't being turned over and they're accumulating ubiquitin tags because they're not being degraded and shuttled, chaperoned out of the sarcomere. And again, just knocking down BAG3 by 50% or 25% at the sarcomere, we get this 40% reduction in Fmax at the sarcomere, looking here functionally. Here on the left are the curves, and on the right here are the individual parameters. Again, quantitative agreement with the human studies, about a 40% reduction in Fmax when we lose about 25% of BAG3 at the sarcomere. Okay, so those studies suggested to us that indeed what we observed in the humans as a correlation was causative because we're looking at just a single change in these BAG3 knockout mice. And therefore BAG3 is in fact doing something at the sarcomere to maintain protein quality control and regulate function. But as we know, an action hero never works by themselves. They always say at the beginning, I work alone, but they always need a team in the end, which is why the Avengers movies make like $1 billion, right? So we have to now assemble the team here. What does BAG3 work with? I also told you BAG3 was a co-chaperone. So it's clearly not doing anything on its own. It does stuff by binding to other proteins. Okay. So we went through all the different proteins that BAG3 is known to bind with, and we were looking to see other things that would co-localize with it at the sarcomeric Z disc. Again, this is back in human cardiomyocytes. So we found that BAG3 is there, like I showed you before. And we found that HSP70 and HSPB8 are also present with it at the Z disc. I want to say when we do these sorts of experiments, we permeabilize them and wash away the cytosol, okay? So this is not saying, I'm not trying to convince you that HSP70 is only at the Z disc. That's not at all what I'm trying to tell you. I'm saying we're removing all that other confounding HSP70 and HSPB8 to see what is at the sarcomere. And so we find HSP70 and HSPB8 co-localized there. And we also did co-IPs here to confirm it. So we isolated the myofilament, and we find here that, of course, as suggested here, that BAG3 binds to HSP70, HSP70 binds to BAG3, and BAG3 binds to HSPB8, this small heat shock binding protein, and HSPB8 binds to BAG3. So we have this complex of HSP70 and HSPB8 at the Z disc, which has actually been known to form before in skeletal muscle. And so it had been called this chaperone-assisted selective autophagy in skeletal muscle was the term that was coined for what BAG3 could do with HSP70 and HSPB8, this small heat shock protein. And we found that this complex of the sarcomere responded to stress, right? We found that the real issue with this, the sarcomeres fall apart when the sarcomere is under stress. And so here we just wanted to say what happens when we put stress on the sarcomere. We just use an MG132 inhibitor, MG132 to inhibit the UPS. And we found in NRVMs here, that's why there's a little dish up here. NRVMs, when you treat with MG132, you have an accumulation of BAG3, HSP70, and HSPB8. So this complex altogether responds to stress in the sarcomere. And of course, we're inhibiting these downstream pathways, so we have an increase in sarcomere ubiquitination as well. We also wanted to know, when you put this complex together, does it require BAG3 or not? What of these three players requires BAG3 or it doesn't? So here we use the heterozygous knockdown mice and also the homozygous knockdown mice to get this sort of almost a titration effect on the levels of BAG3 at the sarcomere. And HSP70 were there at the sarcomere. We get the HSPB8 levels track almost perfectly. The homozygous, of course, it's almost completely gone. And so on the other hand, didn't care. It was there whether or not, suggesting that possibly HSP70 binds HSPB8 binds to BAG3. We also wanted to know if this same thing happened in humans. So we took all of our human data and we just put it into a high BAG and said, what does HSPB8 and HSP70 look like? The sarcomere is highly reliant on the level of BAG3, but we have an agreement here between these two. And so that does require BAG3 to be present. So that raised the question, what does BAG3 need HSP70 for? Like, is this interaction important for it? Because it was high in cancer, right? Remember way back when I first introduced Keanu Reeves, I said, it's also high in cancer. And the concept is if you inhibit BAG3 doing what it's supposed to be doing, you'd have a buildup of and therefore, Jason Getswicky's lab developed a inhibitor of BAG3's interaction with HSP70 called JG98, binding to it. Thus, we thought, well, this would be a great tool for us to use to determine port in the cardiomyocyte. Obviously, we tried this in NRVMs and found that when between BAG3 and HSP70 by about 50%, which we thought was a nice amount. So in this paper, we're trying to look at all sorts of different things. But one of the things that we found was the half-life of BAG3 normally is about 30 hours. And then look to see how long the protein hangs around for. And so you can see half-life BAG3's interaction with HSP70, it drops order half-life when BAG3 and HSP70 are sarcomere is critical for BAG3. Okay, so regardless of the fact on BAG3, clearly all three of these components are critical in order to maintain normal function of the sarcomere protein quality control system. Which brings us to Act 5, of course, of any good action movie. Can the hero actually save the day? Can it rescue the people? And so our question here is, can overexpression of BAG3 in heart failure rescue sarcomeric function, clear out the old misfolded proteins, restore normal sarcomeric function? And what we're going to use here as a model is a mouse model of myocardial infarction-induced heart failure, permanent ligation of the LAD. We allow heart failure to settle in for 8 weeks, and then we give the mouse either an AAV9 of GFP or an AAV9 of BAG3 to overexpress the BAG3 levels. Here showing that BAG3 does in fact get overexpressed in the border and remote zone. And this is showing the injection starts here at point C. We do in fact get a restoration of overall ejection fraction, which is great. But again, we care about the sarcomere. So let's take a look at what's happening to the sarcomere. This is a mouse model, again, of heart failure. I haven't shown you any data in a mouse model, an experimental model of heart failure. I just showed you human heart failure before and the genetic model. So here we're going to look at sham and heart failure, and you can see very similar in magnitude to what we saw in human heart failure, about a 40% increase in the ubiquitination of the sarcomeric proteins in these heart failure mice, indicating depression of sarcomere protein quality control in mouse heart failure. But when we overexpress BAG3, we see at least a partial, significant, but partial rescue in that. We're clearing out some of these misfolded proteins, reducing overall sarcomere ubiquitination. Functionally, you can see here in black is our sham mice, and in red is our heart failure mice, and we see about 40% reduction in Fmax in our heart failure mice. And when we overexpress BAG3, we see a complete restoration of function at the sarcomere once we've overexpressed BAG3 and cleared out these ubiquitinated misfolded sarcomeric proteins. So we can rescue function. But we wanted to know, what is it actually clearing out, right? So it's rescuing function. We're rescuing some of the protein quality control and ubiquitinated proteins, but what is it actually rescuing? And so we took inspiration from Jörg Hofeld's release assay, where he just incubated the cytoskeleton with BAG3. And we wanted to see what was BAG3 releasing from the human sarcomeres who were in heart failure. And so we took human myofilament. Again, remember, these human myofilament with heart failure had increased sarcomere ubiquitination embedded in the sarcomere lattice and a depression in BAG3 levels. So what we did was we took these myofilaments and we exposed them to exogenous BAG3. thinking this is analogous to treating our mice with BAG3, overexpressing it, what is it actually going to be releasing from the sarcomeric lattice? So this will be releasing the clients essentially of BAG3. And sorry, I Googled clients and this was the only picture that came up. So that's what we got. And then we identified those clients by mass spec to say, what is actually being released here? What is this fixing? The first thing we wanted to see was this releasing the ubiquitinated proteins? Because we sort of assumed that it was decreasing it, but is that actually what BAG3 was chaperoning out? And you can see here in these two bar graphs, you can see the blots over there, but in these two bar graphs that the proteins that are being released are ubiquitinated. So we're exposing the myofilament to BAG3 and we're releasing these proteins marked for degradation. So it is in fact chaperoning the proteins out of the matrix we would expect it to. When we analyzed what was being released by mass spec, we found these 8 proteins were significantly increased in sort of the released fraction when we exposed to BAG3. And if you remember, when we did that first mass spec experiment looking for what proteins had high ubiquitination in our heart failure samples, there was that heat map. Many of those proteins were the same proteins that we found that BAG3 specifically chaperoned out of the sarcomere. Thus further connecting this idea that they're marked for degradation, they aren't getting cleared out in heart failure, but BAG3 chaperones them out of that crystalline structure, including, and obviously proteins important for normal function, including alpha-actinin and desmin. We were happy that we saw filament C because this agreed with your Hofeld's group and their previous studies. So sort of confirming our finding, and then also your studies like that was cool that you confirmed our study. Myosin binding protein C and some of these other ones. Now this is sort of like the minimum list. We've actually done this now more times and found there's more proteins, and we've done interactomes for BAG3, HSP70, and HSPB8, and it's actually possibly much larger than this. But from this study, this is what we found, at least these 8 clients. Okay, so to summarize, Sarcomere BAG3, myofilament localized BAG3, decreases in heart failure, in human heart failure and in mouse heart failure. Although it does so more in males than in females. We don't understand that why, except we know it's not an estrogen dependent mechanism. And this decrease in BAG3 of the sarcomere depresses the protein turnover of the sarcomeric proteins, which stay embedded in the lattice structure and causes a decrease in the force generating capacity of the sarcomere and of the myocyte, and thus of the myocardium. This CASA complex, HSP70 and HSPB8 included, has at least 8 clients in the heart, possibly more. And overexpressing BAG3 in the heart was able to restore normal protein quality control of the sarcomere and rescues function. So I have a couple minutes left. I'm going to run through a couple things real quick that I think are cool for the sequel. Now, hopefully, scientifically, the sequel will be better than the sequel to Speed, because Speed 2 was a terrible movie. So BAG3 is involved, actually, I said, in lots of different aspects inside the myocyte. And one of the things that BAG3 also does is it forms this complex here on microtubules and shuttles these misfolded clients to the phagophores and the autophagosomes, and thus to the autolysosomes. And so it's involved not only in removing the sarcomere proteins from the lattice, but also shuttling them to the next stage of the degradation pathway, including the autophagosome. So we wanted to know whether or not this maybe was involved in the dynamic regulation of moving the sarcomeric proteins further along this pathway as well. So what we did was we used neonatal rat ventricular myocytes. If you culture them on pattern cover slips, they get mechanical cues to arrange, and they look more like adult cardiomyocytes. At least the sarcomere looks more arranged. We then expressed a GFP tagged alpha-actinin to delineate the Z discs. And then we used a lentibright to look at where the autophagosomes were going. So here it looks cool, it's they're moving all around, they're doing all sorts of stuff, but it's hard to get very much from this. So let's slow it down and take a look at just a couple of these, so sort of over time. Okay, so here is an autophagosome hanging out. Actually, it's sort of stopped at a Z disc. And we found this really frequently in our live cell imaging. And 3 minutes later, it sort of hops, moves over very rapidly, and it pauses at a neighboring Z disc. And it waits there for several minutes longer before it hops on to the next one. And it hangs out this one for about 20 minutes before it hops to the next Z disc, where it hangs for another couple minutes before it hops directly to the next Z disc. And so there's this pattern of the autophagosomes pausing at the Z discs as they move around, because otherwise they move really rapidly. And we wanted to know whether or not BAG3 was responsible for that or involved in that pathway at all, because it's sort of there shuttling the clients. Perhaps the autophagosomes are stopping at a Z disc like a garbage truck to pick up the misfolded proteins that BAG3 is bringing out of the sarcomeres. And so we actually hypothesized that it might not localize to the Z discs anymore at all because BAG3 wasn't there to bring it. When we knocked down BAG3, so here it's just an siRNA for BAG3, we found it actually didn't stop the autophagosomes from going to the Z discs. So here's one, it's pausing at a Z disc. What we did find though is that once it's there, it just gets stuck there. So this autophagosome is just sitting at the Z disc for 2 hours. It didn't really move around. So that suggested to us, possibly, that BAG3 may not be responsible for causing the autophagophore to form or localize to the Z disc, but that it may be important for loading it up with packages or garbage and sending it on its way. I have one more story about interesting turnover stuff, but I don't want to go too long. And so I'm going to thank my lab. A lot of this work was done by a very talented graduate student, Tommy Martin, who has now moved on to his postdoc in Leslie Limewan's lab. And then the rest of this lab picture is COVID times, so we're sort of spread out here and a little bit smaller than usual. Art Feldman's lab at Temple University helped us with some of the animal models, and I'd like to thank my funding sources, and then also you for your attention, and this funny picture of people trying to fix their car while it's still driving. So I'd be happy to take any questions.